Hi, grade 10s. This is the last stretch. We're going to finish the botany section this week. What I'm going to do with you today is to go through the PowerPoint that kind of brings everything together. And there's also a few new things in there, and it covers the working page on page 74 to 87 of your textbook. There's two short activities that you're going to do in your textbook. We will mark that worksheet from last week. We'll mark that on Wednesday, the 3-page one on plant tissues and organs. We'll go over your cycle test. We'll look at the activities in the textbook, and that will be by the end of this week. All right, so the PowerPoint that I'm referring to is this one. Okay, so it's called support and transport. Support is because of the xylem and the sclerenchyma and all of that. Okay, and the cells being turgid, etc. And then transport is how water moves up, water and salts, and how sugars move up and down the plant to wherever they are needed. So I chose these photographs. This is some kind of veg. It looks like rockets, in fact. I've actually got some growing in my garden. There's a long taproot with lots of little branch roots that are growing from that cylindrical pericycle meristematic tissue in the middle of the root. There's some little axillary buds or buds on the side of the stem that grow into leaves. There's a mealy plant. Mealies don't have a big taproot like that. They've just got fibrous roots, very similar in size. They don't grow deep. So at this first region, that's called a node. You don't need to know that. But these are prop roots, like the guide ropes on a tent, propping it up, making sure it doesn't fall over. And then, this is so bizarre. You cut something down and, okay, yes, it's lovely to look at, but can't grow anymore. All right. So it always comes back to this. Okay, we've learned about the tissues, the meristematic tissues, all right, that then become the permanent tissues, the tissues that fill the plant and strengthen it, the transport tissues, and the covering tissues. So we're always coming back to that. That's groundwork. Okay, lovely diagram here of a section through a root. Got the epidermis, and then all that parenchyma, the cortex, the xylem and the phloem in the middle. All right, and you can see these lovely little root hairs here in the meristematic region for growth and the root cap. And it reminds me of the inside of a carrot. Okay, central steel is actually that region in the middle there where the xylem and the phloem and the other two layers, which I'll remind you about just now. I just love this photograph because of these tiny little root hairs. This is a lentil seedling. Those tiny little things, little legumes that we eat. Root hairs are so small, and there they are as well on another plant. If you pull out a seedling to transplant it and you damage those root hairs, that's why they die, because now you've decreased the surface area for water absorption. So it's always, remember I said, it's always important to take some of the soil with, but you're not disturbing those little root hairs, which are really, really small. And we've seen this as well, of how water moves across the root, through the cortex, through that external layer, right, and then through another layer, that little yellow, those yellowish cells over there, colored in yellow, and then eventually into the xylem and up. All right, I want to show you on another one. This we've seen before. So that other PowerPoint was one that I made for lockdown. And this one, because, we need more diagrams, but this one has got some of those in as well, okay? I'm not going to go through all of them again. And there's that stem that I've been lugging around and doing my gym with. Okay, it's exactly the same one, showing the cross section of a stem or transverse section. And we've talked about how there are separate veins with xylem and phloem, and eventually, when the stem grows wider, when it starts getting ready to make wood, how meristematic tissue in between each of the veins develops, that eventually you've got a ring of meristematic tissue, like this, gone. Okay, that ring there, so there's meristematic tissue, the yellow in the actual veins, and then some parenchyma in between becomes meristematic tissue. Okay, here's parenchyma with chloroplast, there's parenchyma with no chloroplast, just like you see over there. Okay, but it's been stained a blue-green. Okay, that's what color the cells become. So all those different layers going all the way through here, from epidermis, I'm sure this is collenchyma if we had a high magnification, parenchyma going to the middle, there's sclerenchyma, phloem over here. You see a lovely ring of meristematic tissue going through there, so it's getting ready to make an annual ring. And then you've got the xylem, beautiful vessels and tiny little tracheae, okay? All right. Then there, once again, I think this one we have seen, epidermis, lots of parenchyma for strength. There's the vein, there's the sclerenchyma, there's the phloem, here's the meristematic tissue. We don't need to know that it's called vascular cambium, okay, a bit too much detail. And there's the xylem cells, vessels, and just take an educated guess that some of these smaller ones are xylem tracheas, cross section. And you can see all the parenchyma with the little air spaces over here. So this you should really start recognizing this one out. That ring of meristematic tissue, I just really like this diagram. So there you've got the parenchyma on the outside, the cortex, parenchyma in the middle, the pith. There's that ring of meristematic tissue. So to the outside, it's going to make a ring of phloem, and to the inside, it's going to make a ring of xylem, which becomes the wood. So there you go. They call it secondary phloem. So there's the initial phloem in the veins. Now it's made a ring of phloem. There's the meristematic tissue, and there it's made a ring of xylem to the inside. And there's the original xylem. So secondary xylem, primary xylem. Here's the parenchyma. And that's going to happen every single year, so that eventually you're going to end up with another layer of this light pink, which they've used to represent phloem, and another layer of xylem, the wood, to become the annual rings. And there you go. So there's those annual rings again. And we've had a long discussion about that. Beautiful diagram, photograph as well. Here you can see a branch growing out, side branch growing out of the side of the tree trunk. Look at all that bark on the edge there. That's where the phloem is found. You would have seen that in the video that you've just seen. And each annual ring, I mean, look at this. Yeah? You can see when there was lots of water, huge vessels, and there as well. And then the solid wood is not really solid. That's where the cross sections, you'd see the cross sections of the tracheids, the thinner tracheids. So this tree, they said, is 24 years old. All right, and that one you've seen, you don't need to know all those labels, all these over here, All right, and there's another redwood tree. Okay, so now the new wish work, okay? So like I said, some of it you've already seen, we're just pulling it all together. Okay, so from page 78, this PowerPoint summarizes, but you can always go look at your textbook if you want to read it in a bit more detail or in another way. All right, so we know water is continuously absorbed by the plant's roots, and it goes up the stems to the leaves. 2% of that water is used for, what the cell does, photosynthesis, keeping the cells turgid, metabolism. Water is either used or produced during cellular reactions. Okay, think of cellular respiration. Water is produced. Water is used for photosynthesis. Okay, but we know that that 98 or so % of water that is absorbed by the roots is lost from the leaves as water vapor, not liquid water, and it's called, or it happens through the stomata, okay, and we call that transpiration. So also about 10% of water in the cells of the plant evaporates through the cuticle, because the cuticle on the leaves or green stems, it does reduce transpiration, all right, but some transpiration still occurs. Okay, so I know it's a little bit fuzzy, but it's quite a nice diagram because this is what we're trying to figure out now. The water in the soil moves from high concentration to the epidermal root hair cells where there's a low water concentration. That becomes high. From high water concentration to all the parenchyma cells. Next one. Okay, from high to low becomes high concentration. From high to low becomes high concentration. Then all the way up the xylem, then into the xylem, the branch vein, which goes into the leaves, and then a lot of that water gets transpired. 98% is transpired through the stomata. Okay, so this photograph or diagram is quite nice because it shows you, remember, in the root, the xylem is in the middle, that little 4-armed or 3-armed or whatever shapes central core of xylem, which was explained quite nicely in the video. So there the water goes into the root hairs, across the parenchyma, through that first layer, okay, called the endodermis, then through the pericycle, then into the xylem in the middle of the root, and then you end up with separate veins around the stem. So all the way up, you can see how water is moving all the way up into the leaves, into the leaf veins, and out by transpiration. All right, so we need to figure out how this happens. Okay, so This one we've done, the water movement from the root hairs to the xylem. Okay, so like I said, from the soil, high water concentration to the epidermis, either those or the ones with root hairs. Root hairs increase the surface area for water absorption. So now the water is in high concentration in this cell, low in that cell, moves by osmosis or by diffusion. We call it osmosis. Diffusion of water is osmosis. So now the water concentration in this cell is higher than that one. Water moves, net movement of water by osmosis. From high to low becomes high. And then when it gets to this cylinder of cells called the endodermis, the inside skin, because epidermis on the outside, inside skin, endodermis, surrounding where the vein, where the xylem and the phloem is found. Remember, there are these cells. Those cells, okay, over here, the endodermal cells, they're just showing this red line going all the way across the top, but there's the cell, but you can see here, the side walls, okay, are thickened with a fatty material called suberin, if you remember that little story. So water is forced to go through the middle of these cells, okay, which they're trying to show over here, and into the cells on this side, because, into the parenchyma here, because these endodermal cells pump out salts into this tissue, so therefore water is drawn through. All right, so if you just remember that, cellulose cell wall, but the side walls are is an added material called suberin, a fatty substance, so that water has to go through the middle, can't go sideways, whatever. It doesn't want to go near that fatty layer, goes through the middle and into the parenchyma cells over here. But now in that previous diagram, there was another layer here. That first layer inside the endodermis, remember that's the pericycle, which becomes meristematic to make those branch roots, the big roots for extra anchorage. Right, so now, lovely picture of the endodermis again. There's a cell. There's that suberin strip around it. So the water is forced to go through the middle, okay? And yeah, you can see it as well. So the water passes through the middle of the cell to the other side to follow the salt that has been actively pumped out by these endodermal cells. Now, in your textbook, I'm just trying to see here, somewhere in your textbook, it talks about how, oh, there we go, page 83. It talks about 3 ways that water passes across. And I have mentioned before, please don't go and learn all of these. So it was apoplastic, symplastic, and the vacuolar route. It just depends. Water, you know, it takes the easy way out through the cell walls, or through the cytoplasm only, or through the cytoplasm and the vacuoles of every single cell. Please don't go learn that, okay? And then into the xylem and up. Really not interested in that. Okay, so now transpiration, loss of water vapor from the leaves. Okay, so page 78. All right, we have talked about this. So transpiration is a similar diagram if I look on page 78. So there will be lots of water, you know, being brought up by the vein and into the parenchyma cells, the palisades, the spongy cells, and then excess, most of that water vapor, or most of that water evaporates from the cells. You get lots of the water vapor in the air spaces here and in these little chambers just in front of the stomata. So there's a high water vapor content. Water is evaporated out of the cells. So water vapor moves from a region of high water concentration out through the stomata to the outside air. The water vapor leaves the leaves. Transpiration. That's when the, or at least when the guard cells have moved apart and that stoma or pore is now open. Right, so it's all about water moving passively down a concentration gradient from high water vapor content here to lower outside. Right now, what I do normally do is I take a plastic bag, water a plant well, put the plastic bag over the plant, tie it at the bottom. You're actually making your own little water cycle here. Then you would see all the water vapor that's evaporated from the leaves through the stomata condensing on the inner surface, just like, you know, water vapor would condense in the air to form clouds, and then the rain would fall down. So here, the dew, okay, would then be formed on the inside of the bag and go back into the soil, and transpiration, and so on and so on and so on. Okay, so what happens now? So water goes up and into the xylem of the leaves, and then it diffuses from the xylem to those parenchyma cells by osmosis, high concentration to low concentration in the parenchyma cells of the leaves. And then it evaporates from the cells, from the spongy cells mainly, because there are big air spaces between them, and also from the palisade cells, but not as much because they will be photosynthesizing more and using more water. All right, so now there's lots of water vapor in those chambers, and then it diffuses to the outside, which is what that little diagram showed. Now you can actually, they were talking about transpiration pool in the videos, which were really nice. The transpiration pool, in other words, water molecules pulling other water molecules up, it's a really strong force because water molecules stick to each other, and that's why when you pour a glass of water, it comes out in this beautiful flowing stream because the water molecules, they stick to each other. So that's the force of cohesion. Don't need you to know that. And they stick to the walls of the xylem vessels and the tracheates. Adhesion, just like we use glue to adhere something else to paper, whatever, beads to paper, okay? Like when you make those little pictures in grade school. So transpiration pool is the movement of water molecules up the xylem of the stem, etc., into the leaves, pulling more water up. As more water is transpired, more water is absorbed from the roots, as long as there's water in the roots. So there's a continuous pulling of the water column up the plant, but 98% is transpired. All right, so that's what it is. Now, I've got a little thing to show you here. So all I've got, oops, let's see if I can get this out. I've just got a beaker with a few leaves in it as well. Let's just go off stop share. So why are xylem cells so good at transporting water? Well, they narrow and the water molecules stick to each other. And if you look here, there's my beaker of red water. See if I can make that, let you see this. Okay, wait. Let's see if you can see this. Maybe bring it closer to the front. Oops. Can you see that in these tubes, on the left of the screen, a little bit of redness can be seen in the tube, the wider tube. In the middle tube, look how the water naturally moves up that middle tube, which is a little bit narrower. And in the very narrow tube, right on the right-hand side near my ear, can you see how those water molecules naturally move up very narrow tubes? Now that's called capillarity. So the thinner the tubes, like tracheas, or even, xylem vessels are really quite thin, even though they're wider than tracheas, just that force of molecules, water molecules pulling each other up and sticking to the sides of the xylem vessels and tracheas, and capillarity, the fact that liquids move easily up very thin tubes allows water to move upwards against gravity. Okay, so that's one little thing I can show you. Go back. Okay, so where were we? Ooh, go back down. Seems to have gone out. All right, so that's what we're trying to show you here. One back. Okay, so there's even an experiment that we used to do many years ago, but we can't really do it anymore because you use mercury, and mercury is a toxic metal. So if you put that liquid, that heavy liquid metal, into a beaker with a thin tube, and you can see that there's another tube, and then you've got a little twig in this column of water. There's the twig in the column of water. That transpiration pool, water evaporating from the leaves, is strong enough to pull mercury up, because mercury is 13 times more dense than water. So transpiration and pull, water molecules pulling each other up through very narrow tubes, is strong enough to pull mercury. So I remember doing that once or twice, but we don't do that anymore because mercury is actually toxic. All right, now you can actually measure. Now, this is called a photometer. You can measure the rate or the speed of transpiration. So when is transpiration going to be faster? When is it going to be slower? Different conditions in the environment. So what you've got here is a horizontal tube, and it's a calibrated tube. So there's usually some kind of measurements on it, like they've shown you a volume scale. And there's a place where you can put more water in, okay, and another little reservoir here, and a very thin capillary tube. very thin tube, just like the xylem vessels and tracheas. Okay, not as thin, but still thin. You put your little plant in over here, and then as this plant transpires, you could actually measure how much water is lost through the leaves. Because what you do, this little tube here, you lift it out of the beaker, get a little bubble in here, put it back in, and then wait until the water is transpired and pulls through and out the leaves. So you wait until the bubble gets here, and then you time how long it takes for that bubble to move to the other side. So under different conditions, okay, you could place the plant under different conditions, just like it would be, outside in the normal weather, okay? And so this is called a photometer. It measures the rate or speed of transpiration. So the different types of conditions we'll have a look at just now. Here's another one. It's a U-shaped photometer, okay? So you fill it up with water, and there's your little twig. And as the water is transpired, I'm sure you can figure out what's going to happen to this level on this side. So it would have been at the top at the beginning, and you can measure over a certain period of time how much water the level drops on this side because it's been transpired on that side. And then you could put this piece of apparatus under different conditions, like full sun, shady conditions, windy conditions. You can use a fan in the lab or a very humid, condition. If you take, I'm thinking of those big jars we've got, glass jars we've got in the lab, and you put this big jar over the plant, simulating a very humid condition, just like it would be in a forest. Okay, you can see here, you can even push more water in all the way up and then start the whole process again. All right, so that's a simple photometer. So, okay, so we know what transpiration is all about. Okay, it's not wasteful. Even though 98% of the water that a plant absorbs is transpired, it's not wasteful because it helps water with salt to move up to the plant, to the rest of the plant, the leaves, because the salts help to make other things like proteins and fats and DNA and whatever else the plant needs. Vitamins, that's why we eat plants. They make vitamins from the glucose, the sugars that they make during photosynthesis. So the mineral salts are taken up. And just like us, when the plant transpires, it helps to cool the plant. Okay, this you've seen, the two guard cells that move apart. All right, so what affects the transpiration rate? So this is something a little bit new, and there's some graphs on page 79, the little activity that you're going to do, page 79 to 80. So if you think about it, if there's wind, the wind is going to blow the water vapor around the outside of the plant, so therefore more can transpire out, because then the gradient from outside to inside is maintained. If the water vapor stays near the leaves, then no more water vapor is going to leave the leaves, because then there's not a high enough, a big enough gradient from inside to outside. So if water vapor is blown away from the leaves, more can transpire out of the leaves. So wind increases the rate of transpiration. Just like if you're standing really, really hot and you are sweating, if you stand near a fan or in a cool breeze, it will help you to cool down quicker because the water vapor is blown away from your skin. And we know that when sweat evaporates from your skin, how does sweat, liquid water with salt, how does that liquid become a vapor, a gas? Energy, and that heat energy is coming from your skin. So that's why you cool down. Your skin is losing heat because the sweat, the liquid, the water is evaporating, and that uses heat energy coming from your skin. So the same with a plant. The plant will cool down as more water vapor, gets blown away, and then more can leave. All right, so a steep concentration gradient is maintained between inside and outside the leaf. Humidity. So as I was saying, if we put a big jar around this plant, all right, so there now it's very humid around the plant. Like, I remember getting off one plane in Durban. I had to go there for a conference many years ago, and I got off this plane in the middle of April or March and February, and it was so hot. I felt like I was walking into this blanket of humidity. Okay, that's what I think of when we talk about transpiration and humidity. All right, so If there's lots of humid air around the plant, the gradient between inside and outside is not as steep. So less water will transpire from the leaves because the concentration inside and outside the leaf is too similar. So then transpiration slows down. Temperature, well, if it's hotter, water will evaporate faster, so transpiration will increase because the molecules now have more kinetic energy to change from liquid in the plant cells to vapor in the air spaces, and then they pass out through the stomata. Okay, and obviously high temperatures, if it's very hot, then that water vapor is going to disappear. It's not going, it becomes, well, high temperatures decrease humidity in the atmosphere. Not in a forest, I'm thinking. Okay. Then obviously if the temperature is very high from sunlight, you're feeling the heat, light intensity will be very bright as well. So high light intensity and temperature work hand in hand, and therefore the stomata are going to open and allow transpiration to occur. But plants don't open their guard cells when it's really, really hot, like midday and in the heat of summer. They're going to close their guard cells then, because if they transpire too much, then they're going to wilt. All right. So then you're going to do activity on the bottom of page 79, where you're reading off 3 graphs, and it's quite an important activity, please. And you must look at the axes and answer those short little questions on page 80. I'm going to stop this video now, and then I'm going to make a second one.